In this video, we will discuss the maintenance of epigenetic information. So far, we have spent a lot of time talking about how epigenetic marks such as DNA or histone modifications are established, how they can be read, and how they can be erased. What we have so far mostly ignored is what happens when we go through the cell cycle. In order to replicate the DNA, chromatin proteins and histones get dislodged, so that the replication machinery can move through and do its job. How do we maintain epigenetic information such as DNA modifications, histone modifications, and chromatin structure through cell division? We have already discussed how many DNA methylation patterns can be inherited. Let's first go and revisit that process. As you know from your previous discussions, we have two different types of DNA methyltransferases. One type that can add DNA methylation anywhere, the de novo DNA methyltransferases DNMT3A and 3B, and another kind, DNMT1, which preferentially recognizes hemimethylated DNA and is coined the maintenance methyltransferase. Hemimethylated DNA is generated after DNA replication, and so DNMT1 can copy patterns from the old DNA strand to the new DNA strand. Thus, between the DNMT1 class enzymes and the DNMT3 class enzyme, DNA methylation patterns can be maintained as needed throughout development. As we mentioned in class before, this only works for DNA methylation in CpG contexts. The interplay between the two types of enzymes is depicted in a slightly different way in this diagram. Here, we see that the two germ cells that join to form a zygote have very different DNA methylation patterns. These patterns are erased in the pre-implantation embryo, and new patterns are established by the action of the DNMT3 class enzymes. Following establishment, the DNA methylation patterns are maintained by the DNMT1 class of DNA methyltransferases. If patterns need to be changed during differentiation of tissues, this is accomplished by DNMT3 class enzymes adding methylation and by demethylation enzymes like TET proteins and glycosylases. Despite the fact that we have many different kinds of DNA methylation, the interplay between DNMT1 and DNMT3 A and B can faithfully replicate patterns in CpG contexts as needed. We will discuss how other methylation patterns are inherited at a later time. Now, what about histone modifications? What we want is shown here on the right in G1. We want two daughter cells where the histone modification patterns along the genome are identical. But how do we achieve this? First, let's look at S phase. This is where our new DNA strands are synthesized and where we need to package them the same way the parental DNA was packaged. The parental nucleosomes get dislodged, and we need three types of proteins at the replication fork to help us re-establish properly packaged nucleosomal DNA. We need remodelers that can move nucleosomes or evict them as needed. We need chaperones that can bring in new histones to form nucleosomes on the newly synthesized DNA. And we need modifying enzymes that can add whatever histone modification we need. First, let's consider what will happen to our nucleosome during replication. Typically, the nucleosome is split into two H2A, H2B dimers in the H3H4 tetramer. Sometimes, the H3H4 tetramer splits into two H3H4 dimers. If we just focus on H3 and H4, there are basically three options that can happen. First, our parental tetramer stays together and we simply reassemble it behind the replication fork. That means we'll have all the original modifications present and we're done. This is shown on the left. Second, our H3H4 tetramer can split and reassemble behind the replication fork with some brand new H3H4 dimer, giving us a mixed H3H4 nucleosome, as shown in the middle in the diagram. Here, we still need to copy the modifications from the old H3H4 dimer to the new one, and we'll discuss that in just a minute. The third option is that we get a brand new nucleosome that has only new histones, and so doesn't have any of the marks we need to propagate the chromatin structure for. This presents a little bigger challenge. Here now is a more detailed diagram of how the reassembly of nucleosomes after the passage of the replication fork is accomplished. Right in the middle of the diagram, you can see that the parental nucleosome on the left is disassembled, and the dimers are bound to various chaperones such as FACT, ASF1, and CAF1. These chaperones help to bring the histone dimers to the newly synthesized DNA on the right, where they are joined by newly synthesized histones, also brought in by chaperones such as NAP1 at the top, so they can assemble into nucleosomes. As we discussed on the previous slide, depending on how the parental histones are distributed, we can get nucleosomes entirely out of parental histones, mixed ones, or ones made entirely from brand new histones. Next, let's have a look at what actually happens to the histone marks. This model presented here is for silencing histone marks, in particular H3K9 methylation. In mammalian cells, H3K9 methylation is produced by three enzymes: SUV39H, G9A, and CETDB1. H3K9 methylation is read by the reader protein HP1, which has a chromodomain. HP1 also interacts with SUV39H and CETDB1. So, as long as we have some H3K9 methylation present, as in a parental or mixed nucleosome, HP1 can recognize this histone methylation and then recruit the writers, SUV39H or CETDB1, to produce more H3K9 methylation at adjacent histones and nucleosomes. Thus, the histone modification can spread to mixed and new nucleosomes after the replication fork passes through. Similar mechanisms exist for other histone modifications, where a reader protein can recognize the remaining parental modification and then recruit the appropriate writer protein to the site. The writer will act on adjacent nucleosomes or histones and fill in the histone modification pattern as needed. This model represents our current understanding of how histone modification and chromatin structure are inherited through cell division. In summary, epigenetic information has to be transmitted through cell division to maintain cell identity. DNA methylation patterns are maintained through the action of DNMT1 and DNMT3 class enzymes. Nucleosomes are disrupted during DNA replication and reassembled behind the replication fork. The presence of parental histones with post-translational modification allows for the recruitment of modification writers to propagate the chromatin structure.